Okay, we shall continue with the general features of a viral reproductive cycle. Okay, so remember that viruses are obligate intracellular parasites. They reproduce inside the host cells that they have infected. So different viruses would, of course, reproduce a bit differently, but all, most if not all of them, right, do follow certain general steps. Okay, so even though they have different mechanisms and different details in terms of the description of their reproductive cycle, it does follow a general process, so some general steps. Okay, so later, right, you'll be learning about individual viruses. There are like 4 to 5 examples of viruses that you will learn. And you will see that their reproductive cycles are all different, but then they do follow the general steps, right? So knowing the general steps will will help you to better understand and remember, right, the reproductive cycles for the specific viruses. And also when you come across a virus that you have not learned before in the exam, right, then such general features will help you to make it easier for you to understand whatever the information that's given to you in the exam as well. Okay, so the general steps, right, 5 of them would be attachment, penetration or entry, right, replication, assembly, and exit or release. Okay, so firstly for the attachment, right, so viruses must first attach to the specific host cells before they can infect the host cells. Right, so the attachment is specific, they don't anyhow infect random cells, right? It's always specific cells, because on the specific host cells, there would be specific receptors which the viruses bind to. Right? So that gives rise to the specificity of infection. All right, so the binding to these specific receptors on the host cells would be either via parts of the capsid coat for a naked virus, or the viral glycoproteins of the envelope viruses. So a protein that is found on the outermost layer of the virus. Okay. After a bit more, there's a diagram showing you the binding. So in this case, it's an envelope virus. So you have the viral glycoprotein on the viral envelope binding to the specific host cells receptor. Okay, so nothing much. Let's move on. After the attachment, the next step is the entry of the virus. This is a bit misleading, right? So we say entry of the virus, but sometimes it's not the entry of the entire virus, right? So the focus is on the viral genome, because sometimes the virus capsid coat is actually left outside the host cells. Only the viral genome enters. Okay? So even for viruses that the entire virus enter the host cells, right, the entry steps stops when the viral genome has come out into the cytoplasm of the host cells. Okay, so even though we say viral entry, right, it is the viral genome that we are more interested in. Okay, so this part, please take note. Okay, so you will learn about 3 different mechanisms later when we look at the examples of the individual viruses. Right, so I'll explain the mechanisms when we come across the actual example of the virus. So for now, you can just take a look at these 3 and yeah, okay. Then for the replication, it is about the production of new viral genome as well as new viral proteins. Right, so the proteins can come together later, right, to be assembled into the capsid coat, right, and the genome, the capsid coat, and any other proteins can be assembled together, right, into new viruses. Right, so this production, right, of new viral genome or proteins would be making use of the mechanism, the machinery of the host cells. Also the raw materials, right, in terms of amino acids, nucleotides, the ribosomes, the enzymes like RNA polymerase that are needed, right, the energy, ATP as well. So all these would be made use of by the virus. Okay, so once all these viral components have been made, then of course they come together to form new nucleocapsids, right? And the nucleocapsids will be released. If it's a naked virus, then the nucleocapsid is actually the virus itself, right? So then the naked virus is usually released when the whole cells is lysed. Lysed means broken apart. So the whole cells will break apart to release the viruses that's inside the whole cells. For the envelope virus, we have looked at this earlier, right? The nucleocapsid will leave via budding, and then the virus would acquire the viral envelope during this process of budding. Right? So whether it's lysis or budding, right, that will cause the whole cells to die. Right? So when it is lysis, then obviously the whole cells is broken, right? It dies, right? And if it is budding, then you can imagine, right, there are many viruses that are going to be made. And if they all live by budding, then your whole cells would have the cell surface membrane being depleted and gone. So eventually the whole cells will still break apart. Okay, so that is how the whole cells will die. All right. Okay, so this is just a diagram to summarize the 5 steps that we have looked at. Okay.